Welcome back to the deep dive. Today, we've got a really interesting setup. Normally, we have a pile of new articles or a book and we work our way through it.
Yeah, we synthesize it.
Exactly. But today, we're doing the opposite. We're starting with a single point and working outwards. We have this huge stack of literature. Clinical guidelines, WHO documents, pathophysiology textbooks, even some stuff from veterinary medicine.
It's a lot of material.
It is. But the anchor, the thing holding it all together, is just one multiple choice question from a medical board exam.
And it's from 2006. From the FUMARC Dermatology Board, to be specific. Now, 2006 might sound like, I don't know, the dark ages in terms of medical tech.
Right. Ancient history.
But the immunology at the heart of this question, it's actually the direct blueprint for some of the most advanced cutting-edge biologic drugs we have on the market today. It's incredibly relevant.
That's exactly what jumped out at me. This isn't just a memory test. It's a logic puzzle. If you can really, truly understand why the right answer is right and why the wrong answers are wrong, you basically understand the entire mechanism of a disease that affects, up to 20% of children around the world.
That's right. We're talking about atopic dermatitis.
Or what most of us just call eczema.
And that phrase, just eczema, is something that makes dermatologists, a little crazy.
I can imagine.
Your skin. And at the same time, you have something like a civil war breaking out within your immune system.
So that's our mission today. We're going to take this one question apart, piece by piece. We'll use it to explore that cellular battleground you're talking about, identify the main culprits, and really get into the weeds of why the correct answer is the exception.
We need to understand why the immune system decides to attack the skin, and more importantly, which division of that immune system is leading the charge.
We'll be digging into T cells, cytokines, this whole concept of a Th1 versus a Th2 imbalance, and something about a leaky barrier.
It's all connected. The barrier defect and the immune dysregulation are two sides of the same coin.
I've also got some notes here that take us to some unexpected places. I'm seeing French bulldogs and Nobel Prize winners in the outline, so this is going to be a wide-ranging conversation.
It's a surprisingly broad topic.
Okay, let's start at ground zero. The question itself. I want to read it exactly as it was written back in 2006. Let's set the stage.
All right, let's hear it.
The question is, which of the following is not a common systemic immunological abnormality seen in atopic dermatitis? So right away, we know we're hunting for the outlier, the one that doesn't fit.
The false statement. Classic exam trick.
Here are the options. A, eosinophilia.
Okay.
B, increased IgE synthesis.
Right.
C, increased secretion of interferon gamma, IFN-YO, by Th1 cells. And D, increased secretion of IL-4, IL-5, and IL-13 by Th2 cells.
It's a great question because to get it right, you can't just know one fact. You have to understand the whole picture to spot the one detail that's out of place.
Exactly, and we're not going to drag it out. The correct alternative, the statement that is not true about atopic dermatitis, is option C.
Option C, increased secretion of interferon gamma by Th1 cells.
So an increase in interferon gamma is not a feature of this disease.
That's correct. In fact, it's the opposite. In the typical acute phase of atopic dermatitis, the Th1 system and its main weapon, interferon gamma, are actively suppressed. They're sidelined.
And this gets us right to the heart of the matter, this seesaw effect that's mentioned in the pathophysiology notes. To get why C is the answer, we have to understand this rivalry between Th1 and Th2.
You do. The immune system isn't just one army. It's more like a military with different branches, each specialized for a different kind of threat. You've got the Th1 branch, or T helper type 1.
And what's their specialty?
Their job is to fight intracellular pathogens. Think viruses or bacteria like tuberculosis that actually hide inside your own cells. To fight that kind of enemy, you need to activate killer cells. You need to basically tell infected cells to self-destruct.
Okay.
The main signal for that, the general Th1 army, is interferon gamma. That is its signature cytokine. When you see high levels of interferon gamma, you know the Th1 system is at war.
Got it. Th1 fights things inside cells. So what about Th2?
The Th2 system evolved for a completely different enemy, extracellular parasites. We're talking about things like helminths, parasitic worms, stuff that's way too big for a single immune cell to just swallow.
A totally different kind of warfare.
A totally different kind. So it uses a different weapon. The Th2 system releases its own set of cytokines, IL-4, IL-5, and IL-13, just like an option D to recruit a different crew of cells. Eosinophils, mast cells. It creates a type of inflammation designed to expel these large parasites from the body.
And atopic dermatitis, eczema, is fundamentally a Th2 disease.
It is the poster child for a Th2-mediated disease. For reasons we'll get into, the immune system mistakes harmless things in the environment, dust mites, pollen, certain food proteins, for parasitic worms.
And it launches this massive, inappropriate Th2 response.
Exactly. And this is where that seesaw concept becomes critical. Th1 and Th2 systems don't just exist side by side, they actively inhibit each other.
It's called cross-regulation in the notes.
That's the term. The cytokines produced by the Th2 cells, especially IL-4 and another one called IL-10, directly suppress the development and function of Th1 cells.
So it's not just that the Th2 side of the seesaw is up high, it's that its weight is actively pushing the Th1 side down.
Precisely. For example, IL-10 stops other immune cells, like macrophagia, from producing something called IL-12. And IL-12 is basically the fuel the Th1 cells need to activate and multiply. So the Th2 response cuts the fuel line to the Th1 army.
And if the Th1 army has no fuel,
then it can't produce its main weapon, interferon gamma. So in a system that is completely dominated by this Th2 response, you would expect interferon gamma levels to be low, not high, which is why option C is the correct answer to the not question.
That makes perfect sense. And it also explains a really important clinical feature of the disease, doesn't it? The susceptibility to certain infections.
It's a huge one.
If your Th1 system is your main antivirus team, and that team is being benched by an overactive Th2 allergy team, you've got a major gap in your defenses.
You do. And we see the consequences clinically. Patients with severe atopic dermatitis are notoriously susceptible to widespread and severe viral skin infections. Things like eczema herpeticum, which is a devastating infection with the herpes simplex virus.
Cold sore
virus. In a healthy person, it's a nuisance. In a patient with AD, it can spread across all the eczemated skin and become a life-threatening emergency. Same with molluscum contagiosum, another common viral infection. Their antiviral surveillance system is effectively offline.
Because the immune system is too busy fighting a phantom war against dust mites.
Exactly.
Okay, that's a fantastic foundation. Before we go back and analyze the other options, the ones that are true, the notes say the clinic is sovereign. So let's step back from the microscope for a moment. What is atopic dermatitis when you see it in a patient?
Clinically, the definition we use is that it's a chronic, relapsing inflammatory skin disease that is characterized by intense pruritus.
Pruritus, the medical term for itch.
Yes, but I almost feel like the word itch isn't strong enough. In medical literature, we sometimes make a distinction between simple itch and pruritus. The pruritus of atopic dermatitis is relentless. It's life-altering.
And it creates this vicious cycle.
The itch-scratch cycle, a core feature of the disease. The inflammation makes you itch, so you scratch. But scratching causes physical trauma to the skin, which damages the barrier even more. That damage releases more inflammatory signals.
Which makes you itch even more.
It's a miserable, self-perpetuating feedback loop.
And looking at the numbers here, this isn't some rare, exotic condition. This is incredibly common.
It's epidemic, really. The prevalence has, doubled or tripled in industrialized nations in just the last few decades. We're now seeing it in 15 to 20% of all children.
And about 3 to 5% of adults.
Right. And for most people, it starts very early. The vast majority of cases begin before the age of 5.
What's driving that explosion? Is this related to the hygiene hypothesis that we talked about in other deep dives?
That is the leading theory, yes. The idea is that our immune systems evolved over millennia in a world that was, frankly, quite dirty. We were constantly exposed to bacteria, parasites, and so on. Our immune systems were kept busy.
They had real enemies to fight.
Right. And that Th2 system, the anti-parasite system, was particularly busy. But in our modern, sanitized world, with clean water and less exposure to these things, that powerful Th2 system is, it's bored. It's unemployed. And an unemployed army tends to make trouble.
It starts looking for fights where there aren't any.
It becomes overreactive and starts targeting harmless proteins in our environment as if they were dangerous invaders. It's a disease of modernity. We see it far more often in urban areas than in rural ones, for example.
So in a sense, we've traded intestinal worms for eczema and asthma.
It's a trade-off. We've eliminated one set of diseases and inadvertently created the conditions for another set to flourish.
The outline also mentions something called the atopic march, which sounds ominous. It suggests that for many kids, eczema is just the beginning.
It is. This is a hugely important concept. We often see a very typical progression of allergic diseases through childhood. It's like the allergies marching through the body's different systems.
How does it start?
It usually starts in infancy, with atopic dermatitis on the skin. Then a bit later, as the child is introduced to solid foods, food allergies may appear. Peanut, egg, milk allergies.
So it moves from the skin to the gut.
Exactly. Then, as the child gets a bit older, maybe school-aged, the eczema might even start to improve. But then they develop allergic asthma in the lungs. And finally, in later childhood or adolescence, allergic rhinitis or hay fever in the nose.
Skin, gut, lungs, nose. It really is a march.
It is. And there's a fascinating theory with growing evidence called transcutaneous sensitization. It suggests that the initial skin barrier defect in eczema is not just a symptom. It might be the root cause of the whole march.
How would that work?
Well, think about a peanut protein. If your first exposure to that protein is through your gut, your digestive system is generally designed to recognize it as food and develop tolerance.
Okay.
But what if your skin barrier is broken? And your first exposure is a tiny amount of peanut dust in the environment that gets into your inflamed, cracked skin. The immune cells in your skin see it and say, whoa, this is an invader. This doesn't belong here.
And it tags it as an enemy.
It tags it as an enemy. So the skin is actually training the immune system to be allergic to that food. The next time you eat a peanut, your whole system is primed for a massive allergic reaction.
That is a staggering idea. It means that aggressively treating that baby's eczema and repairing the skin barrier isn't just about making them comfortable.
It could potentially prevent them from developing life-threatening food allergies and asthma later on.
That's the holy grail of prevention research in this field right now, isn't it? Can we stop the march before it even begins by fixing the fence?
That's the billion-dollar question.
Well, speaking of the fence, let's talk about the risk factors. The outline breaks it down into genetics and environment. And for genetics, one gene is highlighted in bold, filigrin.
FLG, the filigrin gene. Honestly, the discovery of the role of filigrin mutations in atopic dermatitis is probably the single biggest leap forward in dermatology in the last 20 years.
The name itself, filament aggregating protein, seems like a pretty good clue.
It tells you exactly what it does. Inside your skin cells, your keratinocytes, you have a structural skeleton made of keratin filaments. Filigrin is the protein that acts like rope or glue. It bundles those filaments together, pulls them tight, and collapses the cell down into a hard, flat brick.
And those bricks make up the outermost layer of the skin, the stratum corneum.
That's your wall, your barrier against the outside world. You have a mutation in your filigrin gene, you don't make the protein properly. So your bricks they're poorly formed, they're soft and crumbly.
But the notes say there's a second job that filigrin has, which is just as important. It has to do with hydration.
Yes, and this is the really beautiful part of the biology. As those skin cells, those bricks, are pushed up to the very surface of your skin, the filigrin protein is meant to be broken down.
The body recycles it.
It recycles it into its component parts, free amino acids. And these amino acids, things like histidine, arginine, glutamine, have a very special property. They're powerful humectants.
Meaning they attract and hold on to water?
They literally pull moisture out of the atmosphere and trap it in your skin. Collectively, we call these amino acids the natural moisturizing factors, or NMF. They are your body's own built-in moisturizer.
So if you have a common filigrin mutation, you get hit with a devastating 1-2 punch.
A complete double whammy. First, the structural integrity of your skin cells is compromised, so your wall is weak and leaky. And second, you're not producing your own natural moisturizer, so the wall is constantly dry.
Weak bricks and no mortar.
That's a perfect analogy. And this leads to a clinical state we call xerosis, which is just extreme dryness. And when skin gets that dry, it develops microscopic cracks.
And through those cracks, the allergens get in?
The allergens get in, the water gets out, and the Th2 immune system, which is already on high alert, is waiting.
And that's when the riot starts.
That's when the riot starts.
Okay, so let's zoom in on that riot. This brings us back to the other options in our 2006 exam question. The ones that are true about the disease. Let's start with option D. Increased secretion of IL-4, IL-5, and IL-13 by Th2 cells.
This statement is, for all intents and purposes, the definition of the disease at the cellular level. These three cytokines are the key players. They're the Th2 gang, and they orchestrate almost everything we see in atopic dermatitis.
Let's break down their jobs. The notes suggest IL-4 and IL-13 are sort of the masterminds.
They are very similar, and they share a receptor, so they have overlapping functions. They act directly on the skin cells, the keratinocytes, and they send a signal that does two very destructive things.
Okay, what's the first one?
First, they shut down the production of key structural proteins, including filigrin. So even if you don't have a genetic mutation, the inflammation itself makes your barrier weaker. It's a sabotage mechanism.
So the riot is actively breaking down the walls of the city.
It is. And second, they also shut down the production of antimicrobial peptides. These are your skin's natural antibiotics, molecules like defensins and cathelicinins that kill bacteria on contact.
So they break down the wall and they disarm the guards.
You got it. And that's not all IL-4 does. It has another critical job. It's a primary signal that talks to a different type of immune cell, the B cell.
And what does it tell the B cells to do?
It tells them to start making antibodies, but not just any antibodies. It specifically instructs them to switch their production lines over to making one particular class of antibody, immunoglobulin E.
IgE, which brings us directly to option B in our question, increase IgE synthesis.
It's a direct chain of events. Th2 cell makes IL-4. IL-4 tells the B cell to make IgE.
And IgE is famous for one thing, allergies.
It's the allergy antibody. Its purpose is to attach itself to the surface of mast cells, which are like little grenades scattered throughout your skin and your airways and your gut.
So the IgE sits on the mast cell, waiting.
And when an allergen, say a protein from a desmite, gets through that broken skin barrier and finds two IgE molecules sitting next to each other on a mast cell, it cross-links them.
And the grenade goes off.
Instantly. We call it degranulation. The mast cell just dumps its entire payload of inflammatory chemicals. The most famous one is histamine, but there are many others, and that causes the immediate redness, swelling, and itch of an allergic reaction.
But wait, the notes bring up another cytokine here that's not in the options but seems crucial for the itch, IL-31.
Yes, IL-31. This is a massive breakthrough. For the longest time, we thought the itch of eczema was all about histamine from those mast cells, which is why we gave patients antihistamines.
But they often don't work that well for eczema, do they?
They really don't. And for years, we didn't know why. The reason is that a huge component of the itch in atopic dermatitis is non-histaminergic. It's driven by IL-31.
And where does IL-31 come from?
It also comes from the Th2 cells. But instead of acting on mast cells, IL-31 binds directly to receptors on your sensory nerve endings in the skin.
It bypasses the middleman and talks directly to the nervous system.
It hotwires the itch pathway. It sends a signal straight up the spinal cord to the brain that says ITCH. This is why the itch is so deep and maddening. It's not just a surface irritation. It's a direct neural signal being fired by your own immune system.
So your immune system is literally telling your brain to scratch.
Constantly.
Okay, that covers IL-4, IL-13, and IL-31. Let's look at the last member of the Th2 gang from option D, IL-5. This connects us to our last remaining true statement, option A, eosinophilia.
Right, eosinophils are a special type of white blood cell. You can recognize them under a microscope because their granules stain bright red with a dye called eosin.
Hence the name.
Exactly. Now, in a healthy person, you barely have any eosinophils circulating in your blood. They're a highly specialized force, the heavy artillery that your body saves for fighting parasitic worms.
But in atopic dermatitis, their numbers are way up.
They're way up. And the reason is IL-5. That's its one major job. IL-5 is the cytokine that signals the bone marrow to ramp up production of eosinophils, and it calls them out of the blood and into the skin.
So IL-5 is the recruiting sergeant for the eosinophils. And what do they do when they get to the skin?
They do what they were designed to do. They release the toxic contents of the granules. These are proteins with names like major basic protein and eosinophil cationic protein. These substances are incredibly destructive. They're designed to punch holes in the tough outer shell of a parasite.
But there's no parasite.
There's no parasite. So instead, they punch holes in your own skin cells. They cause immense tissue damage. A lot of the thickening and scarring we see in chronic eczema is the direct result of this friendly fire from eosinophils. eosinophils.
So let's just recap the logic of the question one more time. It's so elegant when you lay it all out.
Go for it.
Option D, increase IL-4, IL-5, and IL-13 is true. That's the definition of the Th2 response.
Right.
That Th2 response leads directly to option B, increase IgE because IL-4 drives that, and it leads to option A, eosinophilia, because IL-5 drives that.
Also correct.
And because that whole Th2 system is so dominant, it actively suppresses the Th1 system. Therefore, you would not see an increase in the Th1 cytokine interferon gamma, which makes option C the false statement and the correct answer.
You've nailed it. That's the entire case in a nutshell.
Fantastic. So now that we understand the why, let's talk about the where. The notes have a section on the anatomotopographic approach, which I love. It points out that the location of the rash changes dramatically with age.
It's a shapeshifter. The pattern of the eczema is one of the biggest clues we have to a patient's age group.
So let's start with the youngest, infants. So under 2 years old. Where does it show up?
In babies, the classic distribution is the face, especially the cheeks and the extensor surfaces.
The outside of the elbows and the tops of the knees?
Exactly.
Okay, why the cheeks? That seems like a strange place to start.
Think like a baby for a second. What are they always doing? They're drooling. Saliva has digestive enzymes and a different pH, which irritates the skin. They're also rubbing their faces on crib sheets on their parents' shoulders. It's a combination of chemical irritation from saliva and physical friction.
And what about the extensors, the knees and elbows?
Again, think about what babies do. They crawl. They're scraping their knees and elbows along the carpet all day. That physical trauma on skin that already has a weak barrier is a major trigger. It's an example of something we call the Koebner phenomenon, where injury or trauma can induce a lesion.
Okay, that makes sense for babies, but then as they get older, the whole pattern flips.
It's a complete 180. In older children and adults, the rash moves from the extensors to the flexors.
The creases, inside the elbows, behind the knees.
Yes, the antecubital and popliteal fossa. Also the neck, the wrists, the ankles, the foldy parts of the body.
Why the switch? What changes?
Two main things: heat and sweat. Those flexural areas are warmer and they trap sweat. Sweat is mostly water, but it also contains salt, urea, and other things that can be irritating if they're held against the skin for a long time. The increased heat and moisture also increase the permeability of the skin, making it even easier for allergens to get in.
The notes also mention a term for what happens in very chronic cases, lichenification. It sounds like something from botany.
It really does look a bit like lichen or tree bark. Lichenification is a sign of long-term chronic scratching. The skin, in an attempt to protect itself from the constant trauma of fingernails, becomes very thick, leathery, and the normal skin lines become incredibly prominent. You see a lot on the ankles or the back of the neck in adults who've had eczema for decades.
There's also a special note here about a specific anatomical location, the eyelids.
Oh, eyelid dermatitis is a huge challenge, both for patients and for us. The skin on your eyelid is the thinnest skin on your entire body. We're talking less than a millimeter thick. This makes it incredibly susceptible. It's very permeable to allergens, especially airborne ones like pollen dust or fragrances from perfumes or hairspray. It's also a nightmare to treat because strong topical steroids can cause thinning and even glaucoma if they get in the eye.
So how do you make the diagnosis? How do you know for sure it's atopic dermatitis and not something else? Is there a definitive test?
This is a really important point. There is no single blood test or skin test that says, yes, this is atopic dermatitis. It is an eminently clinical diagnosis.
Meaning you diagnose it based on what you see and what the patient tells you.
Exactly. We use a set of diagnostic criteria, the most famous being the Hannafin and Rajka criteria. You have to have certain essential features. The number one essential feature is pruritus. It must itch. If it doesn't itch, it's not eczema.
And then you need the typical appearance and distribution we just talked about, a chronic or relapsing course, and a personal or family history of asthma or hay fever.
Right. But we can use labs to support the diagnosis.
That's where the other options from our question come in handy.
They do. If you check a complete blood count, you will very likely find eosinophilia, like in option A. And if you measure the total IgE level in the blood, in about 80% of patients, it will be significantly elevated.
But you have to be careful because high IgE could also mean you have a parasite or other allergies. So it's supportive, not definitive.
Precisely. It's a piece of the puzzle.
What about some of these other clinical signs? These seem like little pearls for diagnosis. The Denny Morgan fold, what's that?
That's a classic. It's an extra fold or crease of skin just below the lower eyelid. It's caused by chronic inflammation and edema, bit of swelling in that area. It's a subtle sign, but it's very common in people with Atopic.
And white dermographism. That sounds counterintuitive.
It's a fascinating physiological quirk. Dermographism means skin writing. In most people, if you take a blunt object like a pen cap and stroke it firmly across their skin, the line will turn red.
Vasodilation, increased blood flow.
Exactly. It's a normal histamine response. But in many patients with atopic dermatitis, the vascular response is paradoxical. You stroke their skin, and after a brief red flash, the line turns white.
It blanches.
It blanches. The paradoxical vasoconstriction. The blood vessels clamp down instead of opening up. We don't fully understand the mechanism, but it's a very specific sign when you see it.
Okay, let's move on to treatment. The therapeutic approach. We've established the two core problems, a broken barrier and a rioting immune system. So I assume the treatment has to target both.
That's the mantra. Repair the barrier, suppress the inflammation. And you have to do both. One without the other doesn't work.
So step one, repairing the barrier. That means moisturizers, emollients.
And lots of them. But it's not just about using any old lotion. We're talking about functional moisturizers. We know these patients are deficient in ceramides, the fatty mortar between the skin cell bricks. So the best moisturizers now actually contain ceramides to physically plug those gaps.
The guidelines say generous use. What does that mean in practice?
It means more than you think. For an adult with significant eczema, we might prescribe 500 grams. It's a big tub per week. The goal is to keep the skin slightly greasy, almost glistening at all times. It's not a cosmetic application, it's a medical therapy. It's corneotherapy, treating the stratum corneum itself.
But when a patient is in the middle of a bad flare, moisturizer alone isn't going to cut it. You have to put out the fire.
Right, and that's where the pharmacology comes in. For decades, the gold standard for treating flares has been topical corticosteroids.
Hydrocortisone, betamethasone, all the things that end in 1 or 8.
Exactly. And they work because they are very broad-spectrum anti-inflammatories. They go into the nucleus of the immune cells and just shut down the production of all those inflammatory cytokines we've been talking about. They tell the whole Th2 gang to stand down.
But they have a dark side.
They do. With long-term use, especially the stronger ones, they can cause side effects. The most well-known is skin atrophy, thinning of the skin. They can also cause stretch marks and visible blood vessels. That's why we have to be so careful using them on thin-skinned areas like the face.
Which is what led to the development of the next class of drugs, the calcineurin inhibitors.
Tacrolimus and pimacrolimus. These were a huge step forward. They work differently. They specifically block the activation of T cells by inhibiting an enzyme called calcineurin. So they stop the cytokine production at the source, but without the risk of causing skin atrophy.
So they became the go-to for sensitive areas like the face and eyelids.
Absolutely. They changed how we manage facial and flexural eczema.
But the real revolution, the thing that has completely changed the game for severe disease, came directly from understanding the science behind our 2006 exam question.
It's a perfect example of bench-to-bedside medicine. For years, we used hammer steroids that hit everything. But once we identified that IL-4 and IL-13 were the specific masterminds behind the Th2 riot,
You could build a smart bomb,
we could build a sniper rifle. And that rifle is a drug called dupilumab.
a monoclonal antibody.
Yes, it's a lab-engineered antibody that has one and only one target. It's designed to find and bind to a specific piece of protein called the IL-4 receptor alpha subunit.
Okay, and why is that receptor so important?
Because it's the shared docking port for both IL-4 and IL-13. By physically sitting on that receptor, dupilumab blocks both of those master cytokines from delivering their inflammatory message to the cell.
It's like putting glue in the lock so the key won't fit.
Perfect analogy. And the results have been transformative. Patients who have suffered their entire lives, covered head to toe in rashes, itching uncontrollably, they get this injection, and within weeks, their skin can be almost completely clear.
And it's much safer than broad immunosuppressants.
Because it's so targeted. You're not shutting down the whole immune system, just this one overactive pathway.
And now there's an even newer class, the JA inhibitors.
The Janus kinase inhibitors. These are small molecules, pills you can take. They work one step downstream from the receptor. When a cytokine like IL-4 or IL-31 docks on its receptor, receptor, it has to send a signal from the cell surface down to the nucleus.
The command center.
Right. And the internal wiring that carries that signal is called the JAK-STAT pathway. JAK-STAT inhibitors cut that wire. They block the signal from the inside. And because they block the signaling for multiple itch and inflammation cytokines at once, they work incredibly fast.
It's just amazing to draw a straight line from a board question in 2006 that asks you to identify IL-4 and IL-13 as the problem to a multi-billion-dollar drug in the 2020s that solves that exact problem.
That's why we do the basic science. It pays off eventually.
Let's zoom out again. The notes have this great section on secrets of the animal kingdom. It turns out we're not the only species dealing with this.
Not by a long shot. This is where the concept of One Health comes in. The idea that human health and animal health are interconnected. And atopic dermatitis is a perfect example.
So who else gets it?
Dogs. It's incredibly common in dogs. We call it canine atopic dermatitis, and it's particularly prevalent in certain breeds: French bulldogs, golden retrievers, Westies.
And is it the same disease?
It is shockingly similar on almost every level. They have the genetic barrier defect. They have the same Th2 skewed immune response. They have elevated IgE. They get the same miserable itch. They're such a good natural model for the human disease that we often test new therapies in them.
So the dogs are helping us find cures for humans.
They are. In fact, the very first monoclonal antibody designed specifically to block itch, a drug that targets IL-31, the itch cytokine, was actually developed for and approved in dogs before we had anything similar for humans.
No way. The dogs got the futuristic anti-itch drug first.
They did. We learned an immense amount from the success of that drug in dogs.
What about cats?
Cats are interesting. They get atopic disease, but it often looks very different. They can get something called the eosinophilic granuloma complex. Remember option A in our question, eosinophilia?
Yes, the buildup of eosinophils.
In cats, you can see the true destructive power of those cells. They can form these huge, angry, ulcerated masses of eosinophils, often on their lips or on their thighs. It's a dramatic illustration of what happens when that cell type goes completely unchecked.
And what about the mice in the notes, this NCNGA strain?
These mice are the living proof of the hygiene hypothesis. They are a spiral inbred strain of mice that are genetically predisposed to atopic dermatitis. But here's the fascinating part.
Go on.
If you raise these mice in a completely sterile, specific, pathogen-free environment, a clean box, their skin is perfectly healthy, they're fine. But if you take those same mice and move them into a conventional animal facility with normal, everyday dusted microbes,
They develop eczema.
they spontaneously develop severe, itchy, inflamed skin, just like a human patient. It's the perfect demonstration that genetics loads the gun, but the environment pulls the trigger.
That's incredible. Okay, let's connect this all to some of the giants of science. The worthy of a Nobel section. How does the story of eczema tie into the Nobel Prize?
You can trace our modern understanding through at least 3 pivotal Nobel Prizes in immunology. The first one would be in 1980, Bananzaraf, Dossett, and Snell. They won for discovering the major histocompatibility complex, or MHC.
Which in humans we call the HLA system.
Right. The HLA genes determine how your cells present antigens, foreign proteins to your T cells. This discovery explains the fundamental genetic basis of who gets allergies. Certain HLA types are just better at showing allergen proteins to T cells in a way that triggers that Th2 response. It's the basis of genetic predisposition.
Okay, that's the who. What's next?
Next is 1996, Doherty and Zinkernagle. They won for figuring out the specificity of T cell recognition. They explain how T cell uses its receptor to recognize a specific antigen presented by a specific HLA molecule. This explains how one particular Th2 cell in your body learns to become an expert at recognizing one specific protein from a ragweed pollen grain.
So that's the how, the mechanism of recognition.
Exactly. And then the most recent and perhaps most relevant for modern therapy is the 2018 prize to Allison and Hanjo.
For checkpoint inhibition in cancer therapy.
Yes. Their work was about taking the brakes off the immune system to fight cancer. They discovered molecules like CTLA4 and PD-1, which are natural off switches for T cells. But in doing so, they gave us the instruction manual for the immune system's braking system.
And in allergy and autoimmunity, we want to push on the brakes, not release them.
Precisely. The principles of immune regulation they uncovered are the absolute foundation for developing the next generation of biologic drugs that can selectively turn immune responses up or down.
It's all connected. The fundamental science of how T cells work is the key to everything.
The immune system is one big interconnected network. You can't understand one part in isolation.
Okay, we're getting close to the end of our deep dive. Let's try to package this all up. If someone listening is studying for an exam or just wants the key high-yield points, what's the T code?
All right, let's do a rapid fire summary. The key takeaways. First, and most importantly, atopic dermatitis is a Th2 disease. If you remember nothing else, remember that.
Got it. AD equals TH2.
Second, the signature Th2 cytokines are IL-4, IL-5, and IL-13. That was option D.
The TH2 gang.
Third, IL-4 drives IgE production from B cells. That was option B. Fourth, IL-5 calls the eosinophils. That was option A.
Right.
Fifth, a bonus one. IL-31 drives the itch by talking to the nerves. Sixth, interferon gamma, the Th1 cytokine, is suppressed. That's the answer to our 2006 question, option C.
And finally, the underlying problem.
The underlying problem is a broken barrier, most famously due to filaggrin mutations, which leads to water loss and allergen entry.
And how would you quickly differentiate this from psoriasis on an exam?
Easy mnemonic. A for atopy and allergy is Th2. Psoriasis is a Th1 and Th17 disease. Clinically, eczema is the itch that rashes, is ill-defined, and it's in the flexures. Psoriasis has sharp, well-demarcated plaques with a silver scale and is often on the extensors.
Excellent. Okay, as we wrap up, we always like to end with a final provocative thought, something to chew on, and the notes point to our old microbial friend, Staphylococcus aureus.
Yes. The chicken or the egg question of atopic dermatitis. Here's the fact. In over 90% of patients with atopic dermatitis, their inflamed skin is heavily colonized with Staphylococcus aureus.
90%.
Yeah.
And in healthy skin.
It's less than 10%. So the staph bacteria absolutely love the environment of eczemated skin. Now, we also know that this bacteria isn't just a passive bystander. It secretes toxins called superantigens.
What do they do?
Superantigens have the ability to activate huge numbers of T cells, non-specifically, bypassing all the normal safety checks. They can pour gasoline on the inflammatory fire.
So here's the provocative thought. For the last 50 years, have we been thinking about this backwards? Is the bacteria just an opportunistic squatter that moves in after the barrier is broken? Or could the bacteria actually be the primary driver of the Th2 inflammation? Are we treating an allergic disease that's actually being caused or perpetuated by a bacterial infection?
If the bacteria are the true villains, then the future of treatment might not be immunosuppression. It might be antibiotics or bacteriophages or even microbiome transplants.
Transplanting healthy skin bacteria onto atopic skin to outcompete the staph.
There are clinical trials doing exactly that right now. It's a complete paradigm shift, moving from immunology to microbiology.
Yeah.
And we just don't know the answer yet.
A fascinating place to leave it. So the takeaway for everyone is check your barrier, use your moisturizers, and try not to scratch.
And remember, if you see Th2, it's not going to be interferon gamma.
Thanks for another fantastic deep dive. We'll see you next time.
Take care.